implanting these electrodes deep within the brain of an animal without causing damage or pain or discomfort to the animal. This was a big step. Now, for the first time, we could stimulate the deep, primitive portions of the animal's brain while the animal is freely moving and awake. Until recently, there was much controversy about what this part of the brain can do. This is called the reticular formation. It's part of the primitive brain. Ted Magoon and Jack French implanted electrodes in the reticular formation of a cat's brain. By stimulating the cat through these electrodes, they were able to activate this small region of the brain. Ed Everts here will show us what Magoon and French found. Ed has implanted electrodes in several different locations in the cat's brain. The cat's been kept awake all night so that he'll be sleepy today. You'll notice he doesn't stir even while we're talking. Ed, would you apply a small electrical current to the cat's reticular formation?
Bob, I'd wake up too if you sent an electric current through my head.
No, you wouldn't, Eric. It would depend entirely on what part of the brain was activated. Now the cat's gone back to sleep again. Ed, would you use another electrode in the cat's head to stimulate another region? You see, the cat doesn't wake up. Now, Ed, would you stimulate once more in the arousal area, please? You see, every time the reticular formation is stimulated, the cat wakes up. When another region of the brain is activated, the cat doesn't seem to notice. By mapping the interior of the brain in this area, Ted Magoon and his associates at UCLA demonstrated that this part of the brain is essential for arousal. In fact, they called it the reticular activating system because this region can activate the whole rest of the brain. It's this part of the brain that wakens us from sleep. We stay awake by keeping this mechanism active. Drugs that make us sleepy and drugs that make us stay awake act in this area. Sleep, wakefulness, alertness, attention, these functions were the business primarily of the psychologist previously. Now we know that certain brain mechanisms are linked to these, and we can work together with a psychologist to explore them. Neil Miller and his colleagues up at Yale University have been studying a particular region in here of the primitive brain.
You've just seen Dr. Everts wake up a cat. He produced a general arousal. I am interested in studying those areas of the brain which seem to have the more specific motivating effects that are necessary for survival. What parts of the brain specifically drive the animal to seek food when hungry, or drive him to flee from his enemies when in danger? Ted Coons and I wanted to find out whether the drive of hunger can specifically be aroused by electrical stimulation of certain primitive parts of the brain. As soon as we stimulate this cat's brain, it starts to drink. But wait, perhaps as in the brain operation you saw, we are only stimulating a motor response, an automatic action. How can we be sure the cat is really hungry? Well, let's move him to one side. You see, he licks the floor. It evidently wasn't hungry after all. The stimulation only caused automatic licking. This shows us that we have to be careful. Brain stimulation can produce automatic activity without arousing a true hunger. This rat has been taught, when hungry, to push through that little door to get the food. Now, when he's not hungry, we'll stimulate his brain. If our stimulation arouses a true appetite, he'll push through that obstructing door in order to get the food. The rat goes through the door to get the food. So, it's not an automatic action. But how do we know that it really is hunger? Maybe it's just a general arousal mechanism that activates whatever habits the animal happens to have been taught. How can we test this? Here's a well-fed rat drinking from a water tube. If we stimulate his brain, and if that activates only general arousal, this rat will just drink faster. Let's watch. You see, he leaves the water and he goes to the food. This certainly demonstrates that stimulation of this part of the brain does arouse a specific drive, which apparently is hunger. But what about a different kind of drive, such as fear? You can readily appreciate that this drive is important for survival. Realistic fear protects us from danger. Jose Delgado, Warren Roberts, and I wanted to see whether we could arouse a drive like fear by stimulating specific parts of the brain. We implant electrodes in a different part of the cat's brain. We make sure the cat is very hungry. We give him food. And as he eats, we stimulate his brain. Watch. He lurches away from his food. But is he really frightened? No. He goes right back to his food after each stimulation. A truly frightened cat won't do that. So all we produced in that cat was a strong automatic action. He was stimulated in the motor cortex. But what will happen if we stimulate another cat in a more primitive part of the brain, an area that elicits fear, either directly or indirectly? Watch. After just one small stimulation, you can see how reluctant he is to go back to his food. He is extremely reluctant. But what does all this mean? I think that these results, along with those of other experiments like them, show that stimulation of certain specific parts of the primitive brain acts in many ways like natural hunger or fear. This is a step toward a better understanding of the mechanism of these basic drives.
I must say, Bob, this suggests a rather negative view of human life, it seems to me, that our behavior is generated by uncomfortable appetites and painful states of feeling like fear.
I think it is, Eric, to a large extent, but that isn't the whole story. Jim Olds, here in a laboratory of the Michigan University, and colleagues of his at McGill and UCLA, have been working with a part of the primitive brain further forward than that under study by Neil Miller. This experiment we're now watching opens the question of whether all behavior is shaped by our needs, discomforts, and fear.
I wanted him to go to the corner marked A. How did I do it? By stimulating every time he took a step in that direction. And instead of trying to get away from the stimulus, he tries to get more. By doing this, I can make him go to any corner I choose. Believe me, we were surprised when we first found that we could control a rat's behavior this way. The experiment you just saw right here was the first step in showing that the brain houses not only mechanisms of punishment, it also houses mechanisms of reward. We found these rewarding areas in the upper part of the old brain. Here, 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 and here.